Welcome, curious minds, to the deep dive. Today, we're really embarking on a journey far beyond our terrestrial home, peering right into humanity's ultimate frontier. Just imagine a future where humans don't just, visit other planets, but truly live there, setting down roots, building societies, maybe even raising families. What's one of the most fundamental, deeply biological, yet profoundly sociological questions we need to answer, maybe even before we launch the first real colonization ship, to make that kind of breathtaking vision a tangible reality. So this deep dive is all about that very question. How do we produce? How do we expand? And, truly establish our species once we've left the comfort and cradle of Earth. Specifically, we're diving into a really fascinating new study. It offers a surprising glimmer of hope, actually, for the future of making babies beyond Earth. We'll be unpacking some groundbreaking insights from an article published on space.com. It details pivotal research from Kyoto University in Japan. They sent delicate mouse cells on this, extraordinary voyage to the International Space Station. Our mission today: to thoroughly explore what this scientific breakthrough truly means for those ambitious long-haul space exploration missions ahead. We need to dissect the monumental challenges that still remain and ultimately understand why unraveling the mysteries of reproduction in space is absolutely critical for our species' long-term future among the stars.
Okay, let's unpack this a bit. Why is the concept of reproduction in space such an absolutely monumental big deal? And why have scientists for decades been so deeply concerned about it? It feels like such a basic, almost intuitive question, right? But the implications as we really start contemplating our future out there in the cosmos, they're truly enormous. Touches on everything, biology to philosophy.
You really hit the nail on the head there. It is primal, and that's exactly why it represents such a critical foundational area of research for us as a species. As humans steadfastly pursue expansion into space, our inherent evolutionary drive to continue the species, to not just survive, but really grow and thrive, that doesn't just disappear. In fact, in the context of space, it becomes, exponentially more urgent. The question of how we'll continue the species remains an absolutely vital one, because the goal isn't just about reaching new planets or, putting up temporary science outposts. It's about establishing a lasting, multi-generational human presence. Think about those truly grand visions that inspire us, right? The bustling moon bases we might build one day, the sprawling Mars colonies we dream of, maybe even those incredible generational starships that could traverse vast interstellar distances over centuries. For any of these really ambitious endeavors to move beyond just, precarious survival and truly blossom into thriving, self-sustaining societies, the continuous arrival of new generations isn't just important, it's absolutely key.
Without the ability to reproduce naturally and crucially safely off Earth, humanity's multi-planetary future becomes fundamentally, you could even say existentially, limited. It just abruptly shifts our whole grand narrative, from one of genuine colonization and settlement to one of mere temporary visitation, or, at best, an extended, potentially precarious stay.
That's a really powerful way to frame it, that distinction between being explorers and becoming true settlers. And settlers, by their very definition, they're the ones who establish roots, build communities, and inherently that includes new life, new beginnings. So, like you said, it's far more than just a biological question. It's this profound sociological and philosophical one. What does it truly mean for humanity to become multiplanetary if we can't actually sustain ourselves through natural reproduction away from Earth? It really calls into question the very definition of a colony or a new home, doesn't it? But, and here's where some might offer an alternative, couldn't we just rely on, advanced reproductive technologies developed here on Earth, like continuously sending up frozen embryos or even, cryopreserved individuals to populate off-world colonies? Why is natural on-site reproduction so fundamentally critical for establishing that lasting human presence you mentioned?
That's a completely valid line of thought, actually. And indeed, those technologies you're talking about, like embryo freezing, they are incredibly powerful tools that we've mastered here on Earth. They certainly have a role to play, probably in the initial stages of colonization, maybe helping to establish the very first generation or providing a sort of genetic safety net. However, the profound importance of natural on-site reproduction for a truly lasting presence, it really boils down to several critical factors. Firstly, logistics. Just imagine the sheer continuous logistical pipeline required to constantly resupply an off-world colony with biological material from Earth. It's incredibly resource-intensive, it's expensive, and crucially, it's vulnerable. Any major event back here on Earth, any disruption to those supply chains, could immediately jeopardize the entire genetic future of that colony. Secondly, and maybe even more fundamentally, is this concept of true self-sufficiency and independence. A colony that relies entirely on Earth for its next generation is, well, by definition, it's not truly independent. It's still tethered, still reliant to forge a new, truly distinct branch of human civilization, one that can eventually adapt and perhaps even, evolve in its new environment. That requires the ability to generate life from within, autonomously. This fosters resilience, robustness. It's moving from just a fragile outpost to a truly viable, maybe even immortal, new civilization. It's about de-risking humanity's multiplanetary future. Establishing these on-site genetic factories, these potential stem cell banks, allows for a sustained continuity, even if, somehow, contact with Earth were severed. And finally, there's the critical element of genetic diversity. A healthy, thriving population needs a diverse gene pool to adapt, to resist disease, to prevent inbreeding over many, many generations. While the initial sample set might be diverse, ongoing natural reproduction within the colony helps maintain and evolve that diversity, maybe in response to the unique environmental pressures of, say, Mars or the Moon. So yeah, while Earth-based tech offers initial solutions, on-site reproduction represents the ultimate strategic advantage for long-term viability, growth, and eventually independent evolution.
That vision of independent evolution, wow, that's incredibly compelling. It really shifts the whole paradigm. But to get there, we have to confront the undeniable truth. Space, as we know it, is an incredibly unforgiving environment, especially for the delicate biological processes we've been discussing. So what are the specific environmental factors in space that scientists have been so worried about when it comes to the human body and particularly our incredibly sensitive reproductive systems?
What's truly fascinating here, and also incredibly challenging, is just how sensitive these incredibly complex biological systems are, even to subtle environmental changes. And like you said, space is anything but subtle. Spaceflight has this broad, pervasive, often negative impact on the way our body functions. And that unequivocally includes our reproductive systems. Scientists are particularly focused on how reproductive germ cells, the fundamental cells like sperm and eggs that carry our genetic information, and their precursor stem cells, respond to what the article calls potentially harmful factors. Primarily, that means prolonged exposure to radiation and the profound absence of gravity, or microgravity.
Right. And these aren't just abstract ideas for the human body, are they? We know from decades of astronaut experience that microgravity, for instance, leads to well-documented stuff like bone density loss, muscle atrophy, and radiation. We hear about that all the time. It's a major, pervasive health risk, something we're constantly trying to shield astronauts against. So going beyond those broad categories, what specific mechanisms might these two big factors, microgravity and radiation, actually disrupt in our reproductive processes, like at a cellular or even a molecular level?
The concern is definitely significant because reproductive cells are, well, by their very nature, they're delicate. And they're crucial for passing on genetic information accurately, without error. Let's start with microgravity. Beyond those effects on bones and muscles you mentioned, which are well known, microgravity subtly, yet profoundly, alters the internal environment of the body. Fluid shifts within the body can change the pressure, the chemical gradients around cells, impacting everything from, say, nutrient delivery to waste removal, even how cells talk to each other, how they signal. For delicate reproductive systems, this disruption could potentially interfere with the precise cellular signaling pathways needed for germ cell development. Or the complex process of meiosis, that's where cells divide to form sperm and egg cells, or even hormone production, which dictates the entire reproductive cycle. Just imagine the intricate choreography of cell division, or the precise hormonal balance needed for an egg to mature, being subtly thrown off balance. These subtle disruptions might not be immediately obvious, but they could have significant impacts on long-term viability or, the health of potential offspring down the line. It's almost like trying to perform a delicate, high-precision surgery while simultaneously experiencing constant, unpredictable, subtle shifts in your operating room. It just makes everything harder. Then you have radiation, which isn't just some vague, major health risk. It's a direct, relentless assault at the molecular level. It causes DNA strand breaks, chromosomal aberrations where parts of chromosomes get lost or rearranged, and point mutations, these tiny changes to individual DNA bases. For germ cells, this damage is particularly catastrophic because it's heritable, meaning any damage or mutation acquired can actually be passed down to future generations. That could potentially lead to increased risks of birth defects, developmental abnormalities, or inherited genetic diseases in any offspring. Even non-lethal damage to these cells could compromise their function or the genetic integrity they're supposed to transmit. And the type of radiation really matters too. You've got solar particle events, these bursts from the sun. They're different from galactic cosmic rays, the GCRs, which come from outside our solar system. Each type has different energies, different penetrative capabilities, posing distinct threats that need different shielding strategies. While our source article mentions muscle loss and arterial health sort of in passing for astronauts, those broader physiological changes really hint at just how profoundly the unique environment of space affects the body. It underscores why reproductive systems, the very blueprint of life, are under such intense and meticulous scrutiny.
Okay, so given all those harsh, really relentless realities of space, the whole idea of successful reproduction up there has, well, for a long time, seemed like a pretty daunting prospect, fraught with uncertainty. But now we have this genuinely exciting glimmer of hope. It's emerging from some truly fascinating new research out of Japan. And here's where it gets really interesting, I think. This research focused on something quite specific, not just, frozen sperm, but what they termed spermatogonial stem cells. Can you help us understand that specific terminology? Why is focusing on these makers of sperm such a potential game changer compared to just freezing a batch of mature sperm?
Absolutely. The researchers from Kyoto University, they embarked on this crucial and incredibly clever experiment precisely to address these fundamental concerns about germ cell viability in the space environment. Their approach was quite innovative and, as you noted, strategically precise. They cryopreserved, which, essentially means they froze using specialized techniques the spermatogonial stem cells of mice. It's absolutely vital to emphasize, just like you did, that these are not the mature, fully developed sperm cells ready for fertilization. No. Instead, these are the precursor cells, the progenitor cells, located right there within the testes. They play this crucial and continuous role in replenishing the supply of sperm throughout an organism's life. Think of them as the self-renewing factories, or maybe seed banks, that constantly produce and maintain the stream of mature sperm. This distinction is critical because, unlike mature sperm, which have a finite lifespan and are terminally differentiated, these stem cells possess remarkable self-renewal capabilities. They offer the potential for sustained production over time.
Okay, so they carefully cryopreserved these factories, these stem cells, specifically chosen for their unique properties. What happened next in their groundbreaking experiment? What did they do with them?
Right, so these meticulously cryopreserved cells were then carefully packaged and transported up to the ultimate orbital laboratory, the International Space Station, the ISS. There, they remained for quite a significant period, a full 6 months. And this duration, it wasn't arbitrary. It was carefully chosen because it allowed for a really substantial exposure to both the persistent microgravity environment and the ambient space radiation that's present in low Earth orbit. This effectively mimicked the challenging conditions a potential long-duration human mission might encounter. It provided valuable data on how these vital cells cope over an extended period away from Earth's protected magnetic field and atmosphere.
And then, after their 6-month stint orbiting our planet, patiently enduring the rigors of space, what did the researchers do when they got them back?
Yeah, so once they were safely back on Earth, the same cryopreserved spermatogonial stem cells were carefully thawed out and then meticulously injected back into the testes of mice that had been specially prepared to receive them. This step was crucial, obviously, for assessing the cell's ability to actually resume their normal function after everything they'd been through. Following this reimplantation, the researchers simply allowed these mice to mate naturally. They just observed them closely for the eventual outcome. And the results, I have to say, were truly surprising and overwhelmingly positive. After a few months, just following natural mating patterns, healthy mouse babies were born.
Wow.
Yeah, it was a tremendous relief for the researchers, I'm sure, and a really significant scientific validation.
Healthy mouse babies. That's genuinely incredible news, especially considering the environment these progenitor cells had just endured for half a year. And the key finding wasn't just that they were born, was it? There was something about the genetics of these offspring that was particularly reassuring. It speaks volumes about the cell's resilience.
Exactly. Even more remarkably, these offspring exhibited relatively normal gene expression. Now, this is a profoundly crucial detail. It goes way beyond just the birth of pups. It means that not only did the cells survive their extraordinary journey and their prolonged time in space, but they also largely retained their genetic integrity and their functional capacity. They were able to produce new life without significant observable genetic abnormalities or widespread transcriptional errors problems in how genes are read in the next generation. The researchers themselves were quoted as being pleasantly surprised. They noted that spaceflight did not affect how well the germ cells sustained themselves through cryopreservation. This finding, it directly contradicts some of the more pessimistic, worst-case scenarios scientists might have imagined for germ cell viability in space. The fact that these cells not only survived, but could successfully produce healthy, genetically normal offspring after being in microgravity and exposed to space radiation for half a year, that's an undeniable significant milestone. It truly suggests a remarkable, perhaps previously underestimated resilience in these specific cell types.
This really is a so what moment, isn't it? If we connect this back to the bigger picture, this particular resilience of spermatogonial stem cells, especially when they're cryopreserved, compared to other reproductive materials we might think about sending to space, it points to a specific biological robustness, something we absolutely need to understand better. It's almost like these cells have some kind of built-in defense mechanism, or maybe the freezing protects them in a unique way, making them especially suitable for space travel.
You've articulated that perfectly. It's likely not just a lucky accident, but rather points towards inherent biological advantages. Spermatogonial stem cells are quite unique. Unlike mature sperm, which are essentially terminally differentiated, highly specialized, and actually quite fragile once they've left the supportive environment of the body, these stem cells possess remarkable self-renewal capabilities and robust DNA repair mechanisms. They are, by their very nature, designed for long-term maintenance and the constant regeneration of genetic material. Furthermore, when they are cryopreserved, they enter this state of metabolic quiescence, a kind of suspended animation. This significantly reduces their vulnerability to cellular damage from things like radiation, because they're not actively replicating DNA or engaging in high metabolic activity. Those are periods when DNA is most susceptible to mutations. So this potent combination inherent repair mechanisms plus a low metabolic state likely confers a superior resilience compared to other, more active or terminally differentiated reproductive cell types. This biological robustness is, therefore, a tremendously promising avenue for future research and potentially practical application. As Mito Kanetsu Shinohara of Kyoto University, the study's first author, aptly stated, and this is a direct quote, "It is important to examine how long we can store germ cells in the ISS to better understand the limits of storage for future human spaceflight." That single sentence really underscores the very practical long-term implications and the next critical steps needed for humanity's journey out there.
So what does this all mean for the big picture, for human settlement on other planets? Are we actually talking about the feasibility of space sperm banks then? Because, like you said, the study focused specifically on these spermatogonial stem cells, continuous source of sperm. That makes a profoundly different impact compared to just freezing a single batch of mature sperm, right?
Yeah, that's a fantastic question, and you've absolutely honed in on the key differentiator. Yes, the idea of a space germ cell bank, or more colloquially, a space sperm bank, is very much on the table. It's becoming a viable and, I'd argue, increasingly essential future application to consider. The successful preservation of these specific spermatogonial stem cells, rather than just mature sperm or, say, embryos, represents a particularly important option for future human use, precisely because it offers the immense potential for sustained natural reproduction over extended periods. It's not a single-use, finite solution. Think about it. If you're establishing a multi-generational colony on Mars, you don't just need to initiate the first family. You need to sustain the ability to keep having families, to maintain a healthy, robust, and diverse genetic pool over hundreds, maybe even thousands of years. Storing these stem cells means you're storing the potential for ongoing, continuous sperm production right there. This material can then be utilized, perhaps for natural conception or maybe assisted reproductive technologies, directly on site, without the perilous and incredibly expensive need for constant genetic resupply from Earth. This approach is absolutely crucial for establishing stable genetic diversity in off-world populations. It allows for a healthy genetic pool that can not only survive, but also evolve and adapt over generations to the unique conditions of an alien environment, like Mars. It truly represents a shift towards biological self-sufficiency. It gives future spacefaring societies the tools to become genetically independent from Earth, eventually.
So it's not just about getting to space, it's really about staying there, thriving there, and fundamentally growing there. That ability to establish a self-sustaining population with enduring genetic diversity. Yeah, that seems paramount for any genuine vision of humanity's future beyond Earth. This isn't just a cool science experiment. It feels foundational to becoming a multiplanetary species. Okay, while this mouse germ cell study is undoubtedly incredibly positive, really exciting news, it's also really important to remember that it's just one piece, right? One piece of a much larger and significantly more complex puzzle. Other reproductive technologies, other stages of development, it seems they face much greater hurdles. This isn't quite a one-size-fits-all solution for making babies in space, is it?
That's an extremely important point. We simply cannot overlook that. And it raises this crucial question about the inherent complexity of trying to replicate life in space. It truly is not a one-size-fits-all solution. We absolutely need a nuanced understanding of the unique sensitivities at each developmental stage. While the mouse germ cell study gives us this powerful glimmer of hope, at least for male reproductive capabilities in space, the article highlights a stark contrast. It says that successful Earth-centered reproductive technologies such as embryo freezing may currently have limited applications in the space environment. In fact, other researchers found that embryonic cells may be particularly sensitive to spaceflight and have problems developing properly.
And just to clarify for everyone listening, the article defines embryos as the youngest form of human offspring, representing the earliest days and weeks of development after an egg is fertilized. So yeah, we're talking about a very early, incredibly dynamic stage of life. Cells rapidly dividing, differentiating.
Right. Precisely. And this stark contrast to the resilience of these cryopreserved spermatogonial stem cells versus the apparent vulnerability of embryos is incredibly important for future planning. It strongly suggests that different stages of reproductive development have vastly varying vulnerabilities to the unique stressors of the space environment. Embryos, with their rapidly dividing and meticulously differentiating cells, are undergoing this incredibly complex, precisely choreographed developmental program. Every single cellular division, every differentiation signal, every genetic expression pathway, it must occur with exquisite timing and accuracy. Microgravity with its subtle fluid shifts, potential for altered gene expression patterns, cellular stress, it could easily disrupt this delicate dance of embryonic development. It might interfere with cellular signaling or essential cell migration during key stages like gastrulation or those gene expression patterns critical for proper organ formation. That could lead to developmental defects or even non-viability. And radiation, of course, would likely be even more devastating to cells undergoing such rapid division, potentially leading to widespread genetic damage and catastrophic outcomes. So while the stem cells show this remarkable, almost dormant resilience when cryopreserved, the delicate, active dance of embryonic development appears to be far more easily thrown off balance in the harsh space environment. It's almost like trying to construct a really delicate, intricate skyscraper during a hurricane. The conditions might just be too volatile for something so complex in its early foundational stages.
That analogy paints a very clear picture of the challenge.
Wow. And the study also points out another important distinction, doesn't it? Something that further solidifies the significance of the Kyoto University research into germ cell preservation. It mentions that arguably more simple procedures, such as freeze-drying sperm itself, rather than the cells that assist in healthy sperm production, may carry health risks for future offspring. What's that about?
Yeah, this is indeed a critical detail that reinforces the value of the stem cell approach. If direct freeze-drying of mature sperm itself carries potential health risks for future offspring, well, it suggests that this process might compromise the sperm's delicate integrity or its genetic material in a way that the cryopreservation of the more robust stem cells does not. Perhaps it's due to the physical stresses involved in the freeze-drying and then the rehydration process. Stresses on the delicate internal structures within the mature sperm, which, frankly, aren't designed for such extreme conditions. Or perhaps there are more subtle changes happening maybe to the genetic material itself, or even epigenetic markers which control how genes are expressed. Changes that only become apparent later, manifesting as health risks in the resulting offspring. This crucial insight makes the method of preserving these precursor cells for later natural production, or maybe even for in vitro fertilization with fresh eggs on site. It makes that method even more vital for safe, long-haul space exploration. It's not just about enabling reproduction. It's fundamentally about ensuring the quality and long-term health of the resulting offspring, not merely the act of bringing life into existence. Safety first.
Okay, so we have a promising lead for male reproductive stem cells, but we're facing significant challenges for embryos and maybe even for directly freeze-dried mature sperm. But what about the other half of the reproductive equation? Female reproductive health in space. That still seems like a significant blind spot, almost like vast uncharted territory in the current research landscape.
It absolutely is. And this represents one of the most substantial knowledge gaps we have right now. It makes the overall picture of human reproduction in space still largely, as the article puts it, uncharted waters. As the article pointedly notes, existing studies are mostly limited to animals and, critically, potentially more limited to men as fewer women have traveled to space. And, traditionally there's often been less research focus on female-specific physiological responses in many fields, including space. While we do have some initial data suggesting that menstruation itself is largely unaffected by spaceflight, which is good to know, there's still a critical and concerning lack of comprehensive data on how microgravity and radiation truly affect follicular development. That's the complex, multi-stage process of an egg maturing within the ovary, and also ovulation, which is the precise release of that mature egg for potential fertilization in human females. These are incredibly intricate processes, governed by delicate hormonal balances and cellular interactions.
And this isn't just a challenge unique to space exploration either, is it? The article brings in a very insightful quote from a space gynecologist, Dr. Varsha Jain. She points out that reproductive health research on Earth is often lacking and underfunded, making the idea of space conception and birth even more theoretical than that. That puts a really interesting terrestrial perspective on this cosmic challenge.
Indeed, Dr. Chain's observation is incredibly pertinent here. This highlights a significant blind spot, not just in space health research, but frankly, in how we've historically approached medical research here on Earth, too. Often with an inherent bias towards male physiology as the default, the complexity of the female reproductive system. From the delicate hormonal balance required for the growth and maturation of ovarian follicles to the precise timing of ovulation, and then the conditions necessary for successful implantation of a fertilized egg in the uterine wall. It presents a unique and multifaceted set of challenges. Each step in this intricate biological process could potentially be highly sensitive to the combined stressors of the space environment, the radiation, the microgravity, the stress itself. Without sufficient targeted data derived from both animal models and, crucially, from human female astronauts participating in focused studies, we simply don't know the extent of these sensitivities. We don't know where the critical failure points might lie. Achieving true multiplanetary human presence necessitates understanding both male and female reproductive processes comprehensively and equally. The current discrepancy in research, therefore, it really underscores a broader issue. An issue of gender representation not just in space itself, but also within the scientific studies that are paving the way for humanity's future among the stars. A fundamental gap that absolutely must be addressed with urgency.
Yeah, it's a stark and really important reminder. Our journey into space needs to be truly inclusive in its research, ensuring we understand the full biological picture for all future space travelers and settlers. If we want to establish diverse, thriving, self-sustaining populations off-world, we need to understand the full biological spectrum, not just, half of it. So this small but, incredibly impactful mouse study, while focused on a very specific biological process, it really seems like a foundational piece of a much larger puzzle, doesn't it? It underlines an important option for future human use for germ cell storage, suggesting that more focused research into these preservation techniques is crucial for safe, long-haul space exploration. What does this really open up for us in terms of tangible future missions and, the long-term viability of off-Earth settlements?
This research is truly a monumental foundational step. It's moving towards achieving human self-sufficiency off Earth, shifting our vision from just temporary outposts to potentially permanent homes. It opens up these profound and exciting possibilities for storing human genetic material, specifically these apparently resilient germline stem cells, not just on the ISS, but potentially on future lunar bases, established Mars settlements, maybe even within deep space habitats on long-duration missions. Just imagine the implications. Future generations could, quite literally, be conceived locally, born on another celestial body, without relying solely on Earth for a constant, vulnerable resupply of new genetic material. This fundamentally strategically changes the whole equation for long-term space colonization. Instead of being entirely dependent on Earth, a dependency that presents immense logistical challenges and inherent vulnerabilities, we could potentially build truly self-sustaining populations with their own inherent genetic continuity and future.
And that's not just about preventing an apocalyptic scenario on Earth, although that's certainly a compelling argument some people make. It's also about proactive growth, right? Ensuring genetic health, fostering true independence for these nascent off-world civilizations.
It's exactly. Imagine a genetic library on Mars, a robust repository of human genetic diversity, safeguarding humanity's future in the highly improbable, but, not impossible, event of a terrestrial catastrophe. Yeah. Or, more optimistically, simply allowing for a richly diverse and adaptable gene pool for generations born away from Earth, free from the constraints of constantly having to import new genetic material. This research brings that ambitious vision a significant, tangible step closer to reality. For you, the listener, this isn't merely about mice in a lab somewhere. It's about the ultimate destiny of humanity. If we want to truly expand into the cosmos, to establish permanent footholds and maybe even entirely new civilizations, we need to know we can sustain ourselves there. Not just as individual explorers, but as an evolving, resilient species capable of reproducing and thriving over millennia. This study provides a crucial piece of that confidence.
It paints a very vivid and, yeah, inspiring picture of what might be possible. But Mito Kanatsu Shinohara, the study's first author, also clearly emphasized the importance of examining how long we can store germ cells in the ISS, sort of hinting at the next critical phase of research. What are the immediate next steps you'd anticipate in this incredibly dynamic field? Beyond simply extending the storage time, what else needs to be explored to move closer to actual human application?
Right, beyond simply extending storage time, which is of course absolutely critical, especially if you think about truly long-duration missions, maybe spanning decades for interstellar voyages. The next steps are incredibly multifaceted. They require a truly interdisciplinary approach. Firstly, researchers will definitely need to investigate the effects of different radiation dosages and spectra, not just the general ISS environment. We need to understand specific thresholds of damage and identify potential, maybe more advanced mitigation strategies beyond just current shielding, perhaps even biological countermeasures. Secondly, we need to look at continuous microgravity exposure versus simulated gravity, like using centrifuges. How does that impact the long-term viability, the function, the genetic integrity of these cells once they're revived? Does artificial gravity, even if it's only partial gravity, offer protective benefits? We don't know yet. Thirdly, and as we discussed pretty extensively, addressing those significant, frankly glaring gaps in female reproductive health research in space is paramount. This includes extensive, carefully designed animal studies focusing on follicular development, ovulation, early embryonic development, all in simulated and actual space conditions. But crucially, as more women travel to space for longer durations, it will also necessitate gathering critical human data on all aspects of female reproductive physiology, from hormonal cycles to the entire process of conception and maybe even pregnancy in space, eventually. We absolutely need to move beyond theory to systematic observation, controlled experimentation, and comprehensive data collection to achieve a holistic understanding for both sexes.
And if we connect this all back to the bigger picture, this deep dive, it isn't just about specific cells or even just about reproduction in a narrow biological sense, is it? It feels like it's about the very definition of what it means to be human in a multiplanetary future. It's about building a truly sustainable presence, not just temporary outposts that always rely on a lifeline back to Earth. It's about our future, writ large, among the stars.
Absolutely. This small, yet incredibly significant mouse study conducted on the ISS provides a vital, tangible piece of that immense, almost daunting puzzle. It offers robust scientific evidence suggesting that at least one major reproductive hurdle, the viability of male germline stem cells after space exposure and cryopreservation, might indeed be surmountable. This really transforms aspects of the dream of human expansion beyond Earth. It moves it from the realm of pure science fiction into a matter of sophisticated engineering, meticulous biological optimization, and sustained scientific inquiry. It fundamentally changes our understanding of what might actually be possible. #outro.
And that brings us to the end of another fascinating deep dive into humanity's most ambitious endeavors. We've unpacked how a groundbreaking mouse study on the International Space Station showed that cryopreserved spermatogonial stem cells, those makers of sperm, can actually survive prolonged spaceflight and then go on to produce healthy offspring with relatively normal gene expression. We've also explored the contrasting, significant challenges that still exist for more delicate embryonic cells and highlighted the pressing, critical knowledge gaps in our understanding of female reproductive health in the unique environment of space. This research, while focused on a tiny, almost microscopic part of the biological process, really has colossal, sweeping implications for humanity's long-term future out there in the cosmos.
It truly does. It pushes us beyond merely visiting space to profoundly considering what it means to truly live and thrive there, to build a future that is sustainable and self-sufficient.
Indeed. Imagine, just for a moment, a future where a child's birth certificate might proudly list Moonbase Alpha, or maybe Mars Colony 1, as their official place of origin. A future meticulously built not just on rockets and habitats, but on the quiet, remarkable resilience of tiny cells sent to space. This pioneering research makes us ask a really profound question. What fundamental aspects of human life, of our very biology and psychology, will we discover are inherently resilient and adaptable in the alien environments of space? And which aspects will demand radical new approaches, ingenious technologies, and perhaps even fundamental changes to ourselves for us to truly become a multiplanetary species? It's a question that will undoubtedly continue to shape and define our incredible journey to the stars. We hope this deep dive has given you a fresh, expanded perspective on the incredible science paving the way for a future among the stars. Keep asking questions, keep exploring, and keep looking up. We'll see you next time on The Deep Dive.